to how that helps clinicians to make diagnosis, accurate diagnosis, and monitor the progression of disease, as well as in the context of research. And when it comes to research, I really want to say that brain imaging studies have really helped us shed light on understanding many aspects of the disease. But I would say that we're still pretty much behind other diseases. So we really have a lot of catching up to do to understand Alzheimer's disease and other types of dementia. All right, so here's I'm going to talk about two types of MRI. The structural MRI, the structural brain imaging, it is a standard MRI scan, which is just showing just showing the anatomy of the brain. This is a picture of the brain. This is a static picture of the brain, and nothing is moving. You're just looking sort of the anatomy. And you use a structural MRI to detect changes in the brain, to detect what we call in Alzheimer's disease atrophy, shrinkage of the brain, changes that are related with the structure of the brain. The contrast to structural MRI, another type of MRI, it is the functional brain imaging, the functional MRI. Functional MRI is a type of MRI where we use to measure brain activity. It is like a movie. Think of it of like a little movie of brain thinking. And when we perform a task, particular brain regions are activated. And when particular brain regions are activated, more blood flow goes there. And the activity it changes. And this is what we detect with functional MRI. And I'm going to show you just a little video here. I hope you can see that. Where you see different areas in the brain becoming more active. So showing that they have different types of different regions of the brain are activated. Functional MRI was actually discovered here at Mass General in 1992, and where they were showing visual stimulus, and they were noticing that the back of the brain, the visual cortex, was activated, and that led to the seminal publication of that discovery.
Dr. Alexandra, can you go back to that slide
Yes.
real quick? Quick question on the one on the right, the functional brain imaging. Um, so is it safe for me to say that the bump in the front is the front of the brain?
Yes, this is the front of the frontal
Oh,
part, the
okay.
frontal, and then this is the back of the brain, as I say, as I show here, the back of the brain, the visual cortex,
Right.
parietal cortex, yes, the back and the front.
Okay. Okay. Thank you.
And so let's talk a little bit about the MRI machine. MRI stands for magnetic resonance imaging. And so what happens when you do an MRI? You can do an MRI for every part of the body, but here we're going to talk about the MRI of the brain. So when you get an MRI, you often change into a hospital gown, and then you lay flat on a table just like this. This is a Siemens MRI machine that we have also in research. And then you're basically slid into the center of this kind of donut hole. And you wear earplugs because the MRI can be very loud and noisy. And the MRI, for those that have had MRIs, know very well that it is also a very tight space. So it can be a little bit claustrophobic, but you're slid into the center of it, and your legs are hanging out of the machine, but your head is actually inside of this hole. And then the machine takes pictures, usually for about anywhere between 30 to 45 minutes on average. And again, it is it is very loud. That's why you're wearing these earplugs or headphones. And we also place a mask over your face. That is called the head coil, and this is what allows the actual machine to take pictures. It's important to remember what is important to remember about MRI is that MRI includes a very powerful magnet. So one of the things that we screen people very thoroughly is to make sure that they are not bringing any metal into the machine. So we want to make sure that when people, before going into the machine, they don't carry car keys, they don't bring other metal. And we do that similar to the way that it is done at the airport with a wand. We just make sure that there is no metal at all, because the machine is very powerful magnet. The MRI has been very common, and it is a safe scan. It is non-invasive, very safe, and very common. And it is so common that even when nowadays, when there is hip replacement or knee replacement and things like that, we use things like titanium, which is safe in the MRI environment. Um, Generally, yes. Mike, you wanted to ask something?
Oh, no, 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 no.
Okay.
Well, actually, you mentioned metal, like going to the airport.
Yeah.
Um, what if somebody has, you always hear about people with metal in their body?
Yeah. So it is, the metal, so they use titanium now because it's so common in the MRI now they use titanium, so there's no problem with the MRI.
Okay.
But other than that, it is a really very safe procedure. So as long as there's no metal in the environment, it's safe procedure. One of the things that we also need to know about the MRI is that there is no radiation at all. There is no radiation that you're being exposed to, and that means that as long as you don't bring a metal into the MRI, you could get as many MRIs as needed. You can get tens and tens or even hundreds of these MRIs without any issue.
Hmm.
Here, I wanted to just show you how, when you do a structural MRI and you get brain images, how the brain images look like, and what we see when we look at these images. And so, when people have dementia, what we're looking for is we're looking at the brain, and we're looking at particular areas of the brain that show shrinkage, atrophy. Shrinkage is another term for atrophy. So the brain, as it is normal for people, as we age, changes in the skin or the muscles, it is also normal for our brain to shrink. But when it comes to dementia, the shrinkage, the atrophy in the brain is way more than just the natural shrinkage that you would expect to see simply by normal aging. And so this is what we look at when we look at the structural MRI. We look at the regions of the brain that have atrophy or shrinkage. And just to orient you here, we have on the left, we have a cognitively unimpaired older adult, and on the right, we have someone with dementia, frontotemporal dementia in this case. And so we're going to look at the brain as those, it is as if somebody face that's looking out of the screen. And so these, I'm going to show you, these are the eyeballs, and that's the
Oh,
front of the brain, and then we're going back to the brain. And so we look at these MRI scans and evaluate the areas of the brain that show the shrinkage. And I'm going to just in the next slide show you what I mean by shrinkage. On the left, you see this cognitively unimpaired individual, and these are the structural MRI images of this brain. And you see that it is a nicely fully packed brain. But you can see here on the right that there's a lot of space
mhm.
between the regions of the brain and these dark holes, which are the ventricles of the brain. And these have expanded. Everyone has holes, the ventricles, but these little cavities in the brain. But in this person on the right that has frontotemporal dementia, these have expanded so that you can actually see them get bigger and bigger because the rest of the brain is starting to shrink.
Okay.
Yes.
So the parts of the brain that are no longer there, do we know what those parts of the brain are used for?
So depends on where exactly the
Mm.
shrinkage happens. A different symptom, different behavior will be affected. And I can talk more about that later on, where I will
Okay,
show you the different patterns of the disease.
gotcha.
Um, yeah, so that's what's happening in clinic. But in research, what we do, we take the structural MRI brain image and we put it into a computer. Then and the computer, we have software now, advanced software that have been developed in a way that they can automatically separate the brain and show us the gray matter, here showing with this salmon pinkish color, the gray matter, which is the outer part of the brain, that it is important for processing information. We with this softwares, we know exactly where the gray matter of the brain is, and we know exactly where the white matter of the brain is. The white matter is the part of the brain that helps communicate between brain regions. And so with these softwares, we can very easily measure the thickness of the gray matter. And the thickness of the gray matter, how thick the gray matter is as you can see here with this uh, uh, green arrows, you can measure exactly, very specifically, how much the gray matter has thinned, has shrunken in someone who has dementia, and that's a very important uh, that's very important for us to know, both for diagnosis and for the prognostication of the disease. So, for example, I will show you here. These are three examples where we have three different types of dementia. And the left, we have all these blue areas that we see here are areas in the brain where they show shrinkage atrophy in frontotemporal dementia compared to healthy older adults. You can see that these areas include frontal regions and temporal regions. In the young onset Alzheimer's disease, the second panel here, you can see atrophy in the brain in different areas. The temporal parietal regions. And then in the late onset Alzheimer's disease, you see also thinning that is happening in different parts of the brain. And so basically, by using these softwares and this processing algorithm with quantitative analysis of the brain structural MRIs, we can identify the common regions that atrophy, that shrink in frontotemporal dementia, and kind of create a signature of shrinkage, a signature of regions that are being affected, that are undergoing thinning, and then another signature for the young onset Alzheimer's disease, and a different signature of regions for late onset Alzheimer's disease.
Um,
It's
um,
really
uh,
important for diagnostic purposes. Mhm.
oh, I got a question from Terry. The question is, when the brain deteriorates, how can you tell how much of their short-term and long-term memory is declining?
So depending on the regions that are being affected, for example, in the hippocampus, we know that when the hippocampus is being affected, like it is reduced, the volume of the hippocampus is reduced, has atrophy, then we know that short-term memory is affected, which is the main symptom in Alzheimer's disease.
Hmm. Hmm.
Yeah.
Okay.
So I want to show you. in this slide here how we use this information, the atrophy, the shrinkage in the brain, to basically to track the progression of the disease over time. This is an individual with frontotemporal dementia. And what we're seeing here is a type of frontotemporal dementia called semantic dementia. What happens is that early on, you have atrophy, shrinkage in the temporal pole, and it is at the very tip of the temporal lobe. Um, but so it as time goes by, and you see that although it starts the atrophy at the tip of the temporal lobe, then it spreads, and this is how we are able to track the progression of the disease. It starts to involve more brain tissue.
Is there a reason
So
why?
one of the studies that we have been working over the past couple of years is that how we can track this progression of the disease over shorter time intervals. So we could look at this. I don't know if you can see here the thinning of this area. For example, we say that there's a couple of millimeters of shrinkage in what we call here temporal horn. This is one of the cavities in the brain, and we see a lot of shrinkage here. And then, we can look at this shrinkage 2 years later, and we see that this has gotten more shrinkage. And sometimes you can see that with the naked eye. Now, what we're doing here, we're really interested in identifying this progression in a shorter time of period instead of saying that, okay, I'm going to look at 2 years later after the first MRI. Now we are getting many, many, many images at the
Mhm.
first visit. And this allow us by getting so many images at the same time, this gives us a more accurate assessment of the thickness at the first time point. And then we are able to repeat that again and see the change in the shrinkage in a much shorter period of time, like 6 months, for example. And this is important. And why this is important? Because if you want, for example, we can see if a drug is working over a period of 6 months, as opposed to waiting to see if a drug is working like after 2 years. So which is really important advancement in the imaging protocols. And now
And
another thing I wanted to mention, sorry, Mike, you were gonna ask
oh,
something.
no, oh, I've got two questions. But one question, and I think you're coming to it, is do they use a dye for you to show this?
A dye? No, this is so here there's no dye. There's no dye. This is not this is a typical MRI, structural MRI. There's no dye involved. There's no more injections involved. Is this the question?
Yes, yes.
Yes, no. Mhm.
Um, okay, so I'm gonna read this. Does does our does our certain lifestyle affect hippocampus brain having shortened memories? These days, and this is a commentary, these days a lot of folks seem to suffer with short-term memory loss. Um, Increasing number of people suffering these days. And can the disease be reversed? Well, we know we've said the disease cannot be reversed. But does
So it so
go
it
ahead.
depends on how much. So as we mentioned earlier, when you talk about dementia, we talk about problems, memory symptoms that affect your daily life, but that are interfering with your daily activities. And so it is there. There are multiple times where, as we age, we may forget things. But if there are hints that make us remember those things, then it's not Alzheimer's disease.
Right.
Alzheimer's disease is when you cannot remember, even with hints, what was said or why you went to a particular place. So it is a disease that it's accompanied, as we're going to see, with particular types of toxin proteins in the brain and particular types of symptoms.
Now, this is a question that comes up all the time. Um, and my answer to this question when it's asked of me is, at what age? The question is, at what age do people start developing dementia? And when I've been asked that question, my answer is there are early warning signs, but you're not going to notice it because so many families come up with reasons why, Nana can't remember this, or why somebody keeps losing their keys. And, they come up with reasons. But, what would be some of the early warning signs that should make you go, uh-oh?
Um, So, for example, if someone will go to groceries and forgets to buy a juice, and you say, "Oh, I thought you were going also to get juice," and they say, "Oh, yes, I forgot,"
Hmm.
then this is not something to worry about. As we age, we all our memory declines. But if on the contrary, you, instead of saying, "Oh, yes, I forgot about the orange juice," you say, you appear to be as if you never heard about this orange juice before,
Oh,
then this is a sign.
that's a sign.
To answer your question about Alzheimer's disease and AIDS, there are
Fast and
the
tight
young
loads. U.S.
onset
forces
Alzheimer's
also hitting back,
disease.
striking launch sites for.
Go ahead. Go ahead.
There is Alzheimer's disease that is what I showed you earlier, and I can show you here with the signature of atrophy of shrinkage in the brain. This is a young onset Alzheimer's disease that we studied and published our work in 2023, where we had 200 patients with young onset Alzheimer's disease. This is a type of Alzheimer's disease that affects people in their 40s and 50s at the peak of their career, earlier than 65 years old. This is a more rare form of Alzheimer's disease. The typical form of Alzheimer's disease, which is the late onset Alzheimer's disease, here at the very on the right,
Hmm.
affects different set of regions, and then it is most common in people over 65 years old. There are different types of dementia, of Alzheimer's disease, and it is not just the age that it is the difference. They have different clinical profile, and as you can see, different patterns of regions that are being affected in the young onset Alzheimer's disease and late onset Alzheimer's disease.
So we got some more questions because the
Um,
chat's blowing up. So here's this one. There's a couple of questions around this. Does anxiety cause or contribute to dementia?
so uh, there are risk factors, and that increase your risk of having Alzheimer's disease. Genetic factors are affecting, affecting Alzheimer's disease. Anxiety may be. Some people with Alzheimer's disease may also have anxiety
Yeah.
symptoms.
Yeah.
Um, it
And
all depends on whether you have the pathology and you have the symptoms, and
yeah.
the diagnosis is, as we mentioned earlier, is a multifactorial evaluation that takes into account all these things.
And Purple Rain, I'm gonna tweak your question. So she
Uh,
asked, is it a myth that certain foods like blueberries, and I'm gonna tweak it by asking blueberry or dark skinned fruits or vegetables can help with memory, such as with the Mediterranean diet?
There have been studies, clinical trials that show that Mediterranean diet can help prevent cognitive decline, improve our not improve our prevent cognitive decline, help us maintain our cognitive function. Whether particular types of food improve particular types of memory, to really answer this question, you need to perform clinical trials. And that's why we're talking here about the value of research to really be able to answer questions like that. Yeah. So I wanted to mention here, if there are no other questions, I wanted to mention here about the value of looking at using functional MRI to look at the brain networks that are affected by dementia. This is an important topic because, as we know, no brain region is an island. Brain regions are connected with each other and they form networks. They communicate with each other. And depending on the communication between brain regions and the network that they form, then we are able to this help us perform different behaviors and functions. And when these brain networks, when there's a breakdown in the communication between in this brain network, behaviors are affected.
Keep going.
And this is, we can only look at that using functional MRI. And so I talked about structural MRI and I talked about functional MRI. And this is a different type of brain imaging, the positive emission tomography, the PET scan. The PET scan has some similarities with MRI, meaning that you have to change to get into a gown. You lay down on a table, the table slits into the donut hole that we mentioned earlier. Your head is inside of it, your legs are out. That's always outside of it. But there are a couple of differences. One is that it is not as loud. It doesn't make as many loud noises. And another one is that it is more of an open space, so it's not quite as tight as the MRI machine. So it's not as claustrophobic. But the major difference between the PET scan and the MRI, from the perspective of someone getting the PET scan, is that you're given an injection in the PET scan. And so what you do, they put an IV into your arm, just like that you've ever gone to a hospital to do a blood draw. And they inject a drug. This drug that is being injected is like a chemical compound that it is tied to a molecule that can act, how can I say that, as it is a radio, it is called a radio tracer, and it can act as a tiny GPS in your body, meaning that it emits radiation, and because it does that, we can, with this powerful machine, we can detect where it is in the body. And we use that to measure the brain metabolism. We use that to measure the amount of toxic proteins in the brain that are associated with Alzheimer's disease. And so I want also to mention that the dose of the drug is very small, so it is considered a safe procedure because it is a very small dose. As you know, Alzheimer's disease, there are two toxic proteins that have been associated with Alzheimer's disease. One is the beta amyloid plaques, and the other one is a tau tangle. The amyloid accumulates and makes plaques in the brain, and the tau accumulates and makes tangles in the brain. And these are toxic proteins, and they are linked to Alzheimer's disease. And so when you perform before doing PET scans, we were only able to see these proteins in microscope. But now with the PET scans or with cerebrospinal fluid tests or blood tests, we are able to measure these toxic proteins in the living brain.
Wow. Hey, looks like we've got a question. Pat, let's unmute yourself and jump in. Okay, Pat, you've got your hand up. Can somebody unmute Pat for me?
I'm asking her to come off mute. I'm hitting the button.
Okay. While we're waiting on Pat, someone's asked a really good question. Is there a link between dementia and ADHD?
I think that it all depends on what are the other symptoms. I cannot really, yes, I cannot really say anything about that.
Okay, all right. Okay. Oh, someone wants to know, is there a side effect to the dye?
It's not a dye here with the PET scan. This is a radio tracer, which is a different thing. It's a chemical molecule that it emits radiation. It is an isotope atom that it is attached to the drug. So there's no dye here, and there's no side effect because this decays very fast. So within a few hours or a day or maybe 2 days, this will go out of your system. So it is a safe procedure.
Okay, cool. All right. Well, I
And I
guess
know that the radiation may sound scary, but if you can think the natural radiation around us in the background, the air we breathe, and the radar with the cell phones or in the airplane, there is a radiation in the background. So we were talking about the amyloid PET scans, and amyloid PET scans are really, really important for the diagnosis of Alzheimer's disease. So I want to mention that up to 30% of cognitively normal people may have a positive amyloid PET scan. So a positive amyloid scan does not necessarily mean that you will have Alzheimer's disease or when the person will have symptoms. You may have amyloid plaques in your brain, but not develop symptoms. And this is because there are multiple factors that play into resilience. And that's what we are studying in our research now to understand what are the factors that make some people having amyloid pathology in their brain, but not develop Alzheimer's disease and not develop memory symptoms, whereas other people have amyloid pathology and develop symptoms. So an amyloid positive scan, when it is accompanied with a cognitively normal testing, so you do the testing in memory and you do very well, you do executive function tests and you do very well, then this does not suggest that someone will get Alzheimer's disease. It suggests that the person has amyloid pathology and maybe in the Alzheimer's continuum, but it is not known if and when the person will have symptoms. On the other hand, if an amyloid PET scan is negative, this strongly suggests that the person does not have Alzheimer's disease. If a person has amyloid positive PET scan and has memory symptoms and all these other symptoms that are associated with dementia, then this is Alzheimer's disease type of dementia.
So if a person, so I'm looking at the third bullet. If it comes back positive, how often should they get tested?
So if it becomes positive, and if they are constantly forgetting things, and the family is really worried about the memory,
Mhm,
then they should
mhm.
get tested. They should go. They do poorly on memory tests, then they should. Um, I would assume that the test would be done in the first neurological evaluation,
There you go.
the
So,
best,
so,
the first evaluation with the doctor. Yes.
so Alexandra, I want to go back to, and you don't change your slide, but remember when we talked, you mentioned earlier about basically a memory diagnostic workup,
Mm-hmm.
and you talked about all of that. Um, we see on the news all the time now about a certain individual who brags about getting a cognitive test. A memory diagnostic workup isn't it a little bit more than being able to remember four animals and what time it is, things like.
It is definitely more complicated. It includes a variety of neuropsychological tests that are aimed to measure and tap into different types of memory and processes that are being involved.
And I assume it takes more than a half hour.
It depends on the evaluation, yes, but it's
Okay.
not just one test for sure. Um, I wanted to show here just to bring together the structural MRI and the PET scans and really show you one example that shows of someone who has Alzheimer's disease. You can see here with the structural MRI and the red arrows, these are the areas you see the blackness, the holes. These are the blackness, the areas where it shows shrinkage, and it is at the back of the brain. You can see that. Now, in the one type of PET, which is called FTG PET, that uses glucose and measures the metabolism of the brain, you can see that exactly at the back of the brain, and these exactly regions, there is not much metabolism going on. You see that reflected by the green as opposed to the red of the brain, the red here that shows that there is activity and metabolic activity. Here, at the same areas where you see degeneration, shrinkage, atrophy in the brain, you see that there's not much metabolic activity. And then in the PET scan, in the tau PET scan, you can see that these red areas are exactly the same areas as where the shrinkage is happening, the hypometabolism is happening. You can see here that these are the traces where they go and they bind to the toxic tau protein that we mentioned earlier. And then at the very end, here on the right, you see the amyloid PET scans. That here we don't see at where the amyloid plaques are. This is just whether someone has amyloid plaques over a particular amount, over a threshold. So this is a positive amyloid PET scan, and that shows hypometabolism in particular areas of the brain, and where you see tau tangles and atrophy in the same areas, and this is suggestive of someone having Alzheimer's disease. So I wanted to say that we really need both a structural MRI and the PET scans to make an accurate diagnosis. So in terms of the research application of PET scans, an amyloid PET scan can be used to include or exclude a person in an Alzheimer's medication study. So we know there are a couple of new medications that are these antibodies that are directed against amyloid in the brain. So these are the little antibodies that are being injected. They go into someone's brain and they latch onto the amyloid, and then they pull it out and they excrete it out of the body. So for all these studies, in order to be eligible to go into the study, you need to be amyloid positive. So it is really important to undergo an amyloid PET scan. It's an invaluable way of allowing us to know who should be included in this study and who should not. Another thing I wanted to mention is that the tau PET is also correlates, as we saw earlier, with the degeneration. It correlates with the shrinkage that is happening in the brain. And so we can use the tau PET and the MRI to track progression of the disease across time. And in fact, in many of our research, we are developing models that we combine MRI and structural MRI
Oh,
and PET scans to predict when or how the clinical decline is going to happen, or when someone will progress to dementia. One of the most frequent questions we get in the clinic is, when will I lose my independence? And the time from someone that has mild dementia, mild cognitive impairment to progressive dementia is important to know. And we have been working on understanding and predicting this time by using both MRIs and PET scans. And so these provide complementary information that the neuroscientists, use to be able to predict with more certainty the time of progression to dementia. And then we also have, blood tests that can detect the toxic proteins associated with Alzheimer's disease, and also blood testing information can be used with the MRI and the PET scans to better inform the diagnosis and the prognostication of the disease. And finally, we are also developing new radiotraces, new drugs that allow us to evaluate things like inflammation in dementia. We get a sense of what is happening from an inflammatory cascade point of view. So there are a lot of questions we can answer by brain imaging, both structural MRI and PET scans, to better understand the disease. And so I think, but the technology is there, advancement is happening as we speak. The algorithms are being developed to predict to help us make a more accurate diagnosis, to help us predict the progression of the disease. But we really cannot do much if people do not participate in our studies. We have funding from the government, we have the technology, and it is really important for people. None of the things that we know, we would know if there were not studies, and if there were not, you, the volunteers who come to our studies. And I think it's also important, when you think about research and participation in research, to also think about, volunteers that come from diverse backgrounds, because I have to tell you that when you get a result and it comes from a particular group of people, it's not that accurate to say that this generalizes to different types of populations, or to people with different with diverse background. So it is really important to have participants in our studies that come from diverse background, to allow us to have more valid, more accurate results, also from different age spectrum. So I really wanted to thank all of the participants in our studies, and reiterate how important it is and how grateful we are for all of you who participate in our studies and help us better understand the disease, because at the end of the day, it's not just the science, it's about the people and the humanity, and we really want to, with you, to partner and understand the disease. And I know that there's the MADRC, so in the Massachusetts Alzheimer's Disease Research Center, we have multiple studies, and here I've just listed the memory and aging research study that it recruits participants over 60 years old and over with or without memory symptoms. But there are also additional studies for those that are really interested in getting more involved in our research at Massachusetts Alzheimer's Disease Research Center. Please sign up here at the registry MADRC and join our research registry and help us learn more about the brain health. I think it's really important. And if we have time, Mike, I would love to show you more about some exciting findings that we have from our own research that we have benefited by participation from the community and from people here in Boston area.
You were reading my mind, and we have plenty of time. Don't worry. But I've got some interesting questions. You kind of already answered it with this slide here, so let's leave that slide there. But someone's got me, they're saying these MRIs seem to be lengthy in time. So, and
Oh,
they said,
yeah, that's a great question. 30 to 40 minutes. So it might probably in our protocols, usually it's 30 minutes, but the whole process might take 45 minutes because you need some time to change the wand that we mentioned earlier just to make sure there's no metal. And then the actual protocol, it's about 30, 35 minutes. And then 15 minutes to get out and the whole product, I would say 45 minutes to 1 hour, but the actual protocol, you being in the scanner is about 30, 35 minutes.
okay. And I want to thank Haley Sharp. Haley's doing a great job with some of the questions. Okay, yep. And okay, so you were saying, oh, hold on, I lost myself. I'm back. So you were saying about some of the folks that have some of the experiences of participating in research.
Yes, I was going to just share with you how participating in research has helped us towards better understanding healthy aging, successful aging, what we call super agers, and then how that helps us also now try to develop a new treatment for Alzheimer's disease that we run in clinical trials in the Massachusetts Alzheimer's Disease Research Center. So if there's time, I'd be, and if there's interest, I'd be happy to go over these findings.
Let's go. We got an hour more.
We have an hour?
We got plenty of time. Let's rock.
Okay, that's great. So maybe then can I change the presentation?
Sure.
So that I can show you the other sides?
Sure. Perfect. I'm just gonna say, don't ask me to do it because I don't know how. I openly admit that. Everyone knows I had children to show me how to use technology.
Did I stop share?
Yes, you did stop sharing.
Okay. All right. Please give me 1 moment.
Okay. How about preventive things besides social and food? Thanks, Haley. Like I said, Haley's rocking in the chat. She's rocking in the chat. And I just want to say, in case anybody didn't notice, we are at 179 folks. As always, I want to thank everybody for hanging out with us. Instead of putting your email address in the chat, I know Haley is gonna put our email address. And Alexandra, when you get a chance, could you put your contact information?
Yes.
So that in the chat.
In the chat.
See, Haley just put MADRC down there. So if folks want, please, reach out to us. If you weren't able to do the QR code, you can email us and we'll make sure. Oh, see, look, we were reading my mind again. Because that's one of the questions that was in the chat, which I was going to get to. Is it common for memory to decline as people age?
We'll see that it's common, but not inevitable.
It's
Um,
not. Yep.
It's not inevitable. Okay, so this is my email address. But perhaps we can also share the registry, the sign up
Yeah.
registry.
Well, you had the slide with the QR code for the registry. So when we wrap up again, if possible, let's swing back to that slide.
Absolutely, absolutely.
Okay. But this is, someone put in a really, it's an analogy I've used, which is the older we get, the more files our brain has to memorize,
Mhm.
which why it takes us longer. I mean, I'm notorious, and everyone knows this, who knows me. I'll
We
hear a
know.
song, I know all the lyrics to all that song. I just can't remember who sang the dang thing. And I will be thinking about that all day long. And then like midnight, it'll be like, oh, Johnny Mathis. So I've come to the conclusion it's because my brain is still looking for that song, whereas I'm thinking somebody with some type of dementia, they may start to look for it, like with the grocery store thing. You go to the grocery store to buy orange juice, you get to the grocery store, and then you can't remember what you went there for. So you come home with mangoes. So, you know, so as
It
you
is,
get older.
we all well know this. It is common for memories to decline as people age, and this is an illustration of this change. We see that significant memory starts to decline as at approximately age 60. And here are data from one of our studies on memory. Looking at this average data in isolation, this is normal aging, this is young adults. Looking at this data in isolation, one might come up with the conclusion that memory decline is an inevitable consequence of aging. But let's take another look at the same graph with a closer examination of individual differences of these individuals, because it is really important to, and I'm taking some advice here from Hippocrates, who said that it is really important which person the disease has than what the person the disease than what the disease the person has. So it is really important to look at each individual. And so let's look at the same graph that I showed you earlier. But now, with a closer examination of these individuals as represented here with these data points. If I can direct your attention to these individuals here on the right that are half circled, you will notice that there are some older adults that show memory capacities really rivaling those of young adults. You can see that they're at approximately the same level. These are older adults and these are young adults, but they are at the same level. So now I'm going to say that memory decline may be common, but not inevitable. There are some older adults that actually perform as well as young people on memory. And we call these older adults superagers, successful agers. And this is a more recent term, but as we go along, the term of superagers and successful agers existed before it became, long ago, before it became popularized. So back in the lab, what we define superagers as those older adults over 65 years old who perform as well as young adults in their 20s, in the age of many of our students. So they have memory as good as young adults in their 20s, and they perform normal in executive function. How do we screen them? We give them a memory test that includes 16 unrelated words. And then we ask them to recall these words 30 minutes later. An average 50-year-old will remember about 9 words. Average 80-year-old will remember about 5. Superagers, successful agers, remember them all, just like young adults.
Wow.
And Yeah, so it's pretty amazing, but we are neuroscientists, and the first thing we wanted to do is to use brain imaging to understand the characteristics of the brains, the features of the superagers, the brain features of the superagers that help them have resisted the memory decline that comes with aging and accompanies aging in most of us. And so we were very interested in pinpointing what is neuroanatomically different in their brains compared to their peers and compared to young adults. Before I show you how a superager looks like, let me show you how the brain of a healthy, normal, typical older adult over 65 years old looks like when compared to an average young adult in an MRI scan. As we mentioned earlier, changes in the skin and the muscle and other
Mhm.
parts of the body. Shrinkage in many brain regions is thought to be a normal part of the aging process. And so here with the algorithms that we mentioned earlier, we have quantified this shrinkage where we had this group of older adults, this is the right and the left hemisphere of the brain, a group of older adults, and we compare them with the group of young adults. As you can see here, all these yellow spots are areas in the brain where healthy typical older adults have atrophy compared to young adults. And so this is an age-related atrophy in both the right and the left hemisphere. And you can see that it is pretty widespread effect. It affects all these yellow spots or areas of the brain that have shrinkage, and it is quite many. Now look at the brains of superages compared to young adults. You see that they are not yellow spots at all. And remarkably, the brains of superages are truly youthful. And when I say youthful brain, I mean they were statistically distinguishable from young adults. And if you were to present me two brains in MRI scans, one from a young adult and one from the superager, and looking at the part of the brain that are important for memory, I wouldn't be able to tell you which is which. So they have maintained really youthful characteristics. It's like a 25-year-old brain in a 75-year-old body.
So, Alexander, I got to jump in. Go back. Because my grandfather was one of them super age people
Mhm.
because he passed away somewhere between 105 and 100. He lied about his age. He had two birth certificates. He outlived two and a half wives.
That's
And
a great point. Before you continue, I want to make a comment here.
go ahead.
It's and thank you for mentioning that. So when we talk about how we define superagers, so superagers there are superagers, the centenarians, that are known because they for their extreme age, they are over 100 years old. What we're talking here are superagers over 65 years old who are well known for their exceptional memory.
Hmm.
They may not be over 100 years old. We define them as long as they are over 65 years old and they have memory like young adults, we define them as superager. So having said that, what was your question?
Well, my question, and this is something that is asked I'm surprised no one has asked it yet. Is it hereditary?
Oh, we can talk about that. So it is a mix, yes. So that I was going to just say that exactly in my next slide, Mike. Um, we're getting closer to understanding. With brain imaging, we do a lot of brain imaging on superagers. We do the structural MRI, where we look at the structure of the brain that I showed you earlier. We also used functional MRI that we mentioned earlier that looks at the function of the brain, how the brain is activated, and we found many differences in the superages and the typical older adults. The superages tend to be their brains are more connected, functionally connected. Their brain regions communicate more efficiently between them compared to their peers. And when they perform a task, the superagers they're using strategies to help them remember what they learned. So it's not that they're working harder, but definitely they're working smarter. They are using different strategies to help themselves remember the material. But so with all this brain imaging that we're using, we are getting closer to understanding what is super in super ages, what helps them be resisting the memory decline. But we don't know yet how someone becomes a superager. There are many unknowns, but it is almost certainly a mix of genes, one's environment, and brain health behaviors like staying mentally, physically, and socially active that we believe allow superagers to resist the decline in brain power. Superagers come from all walks of life. One common characteristic of superagers is that they are very social, they're very connected, they report stronger, closer, tighter family connections. So social factors definitely play an important role. And there are probably numerous other factors that we have not yet discovered. So just to conclude our discussion on these super sharp superagers, I thought I should let the superagers speak for themselves. These superagers have been the focus of several pieces from the media, including this one from BBC. It features superagers giving us advice on how to stay young. And I think you will see from this that it is as much about your attitude towards aging as it is your brain health. Let's see if it plays. Does it play okay?
Yep, there you go.
Don't lose in life. Just keep busy and keep moving. You need to find something that you love that makes you feel curious and alive. It's a number to me. I feel much younger. I started exercising very young. And when I say young, in my 50s.
I just think I can accomplish wonders. I like to do things. I don't want to sit in the background. I'm an optimist first and foremost.
Have variety in your life. Enjoy what you're doing.
Enjoy being challenged.
Don't take anything for granted, including your own body. Make new friends, try new things, live the life you love, and love the life you lead. Just keep your mind open and be as argumentative as you want to be. Feel like I'm the last one standing.
Alexandra, I gotta share this. Central Boston Elder Services every year holds a celebration honoring people who have turned 100 years old. And it is I've gone for years, and it is amazing, and it's an amazing event. It's as I always say, when I grow up, I want to be them. So go ahead.
When we meet elder adults, a common question we get is, how can I make myself a superager?
There you
How
go.
can I prevent cognitive decline that happens in normal aging? And in this day and age, probably the biggest question they ask is, how can I prevent myself from developing dementia, like Alzheimer's disease? And I think in addition to normal aging, superagers and the study of superagers can also help us develop effective treatments for Alzheimer's disease. And I'd like to show you very briefly an example of such treatment. It is using an innovative technology, non-invasive brain stimulation, that we have been recently developing, which was inspired by superagers. And it is a personalized treatment for patients with Alzheimer's disease. And this clinical trial, we used brain imaging and to identify the features of the superagers. And then we now know that there are particular brain areas in superagers that predict their sharp memory. So this information points us to targets for treatments and interventions that induce neuroplasticity with non-invasive brain stimulation in older adults to effectively mimic, I would say, the resilient brains of superagers. And since 2019, we have successfully launched 4 clinical trials funded by the National Institute of Health that use this non-invasive brain stimulation technique as a treatment for Alzheimer's disease. And this is part of the MADRC, the Massachusetts Alzheimer's Disease Research Center. So in these trials, we induce neuroplasticity to improve the function of brain regions that are important for memory. And by strengthening the function of these brain regions, we mitigate the memory symptoms of Alzheimer's disease. And let me share with you the results from our first patient. This is a patient with a typical form of Alzheimer's disease. It was a patient that came to the clinic that had Alzheimer's disease. He could not remember what, like he had language impairments. So his main symptom was he could not remember words. So when he came to the clinic, this is how his performance looked like in a standardized test of naming. As you can see, the patient improved, shown here in red, his ability to remember words after stimulation, shown here in red, but not after placebo, which is the absence of stimulation, shown here in blue. So patient showed an improvement in symptoms after stimulation. And here's how the stimulation affected his brain function. When the patient came to the clinic early on, communication between brain regions that we talked about that we were able to detect, to measure using functional MRI, the communication between brain regions looked weak. But then after stimulation, we found the communication between brain regions became stronger compared to baseline, as you can see. So the results are really preliminary, coming from one patient, which we published 2024 in Brain Stimulation Journal. But they are very encouraging, suggesting that there are plasticity effects happening after stimulation. Our trial is ongoing as we're trying to better understand the efficacy of this treatment. But I hope I show you today the potential to use brain imaging findings from healthy older adults, successful agers, and translate them for direct benefits for patients with Alzheimer's disease.
So, someone's going to ask this question, if not today, some other day. Is this study still enrolling?
Yes, yes.
All right. So anybody who wants to know, the information is in the chat, but we'll make sure we put it in there again. Also, it's funny because I've got a question. There's been a couple of questions and comments about stimulating the brain through puzzles, crossword puzzles, Sudoku. Is that the same as what you're talking about here about stimulating the brain?
No, it's not the same. Well, let me clarify that when the brain is passive, the brain is not active. When the brain is engaged and when we're performing particularly challenging tasks, we are engaging our brain and plasticities, and then new connections are being formed. So we have what we say neuroplasticity when we perform challenging tasks. This is a different type of of stimulation. So in this case, we have a magnetic coil that induces electrical current non-invasively in the brain. And by doing that, we are strengthening the function of neurons and their connections to other neurons in this way. So there is not learning involved, but we know that with learning and engagement in challenging tasks, there is new connections are being formed. You observe plastic effects in the brain.
Oh,
I also want to make sure that some people say that they would some people have linked computer tasks and games to prevent cognitive decline. I would say that in a neuropsychological testing, it is and training. This is a more of a structure, more of a challenging training that it is involved that we have seen positive benefits. Yeah, it is more structure, I would say.
okay. All right. From super agers to Alzheimer's.
Yeah, so I wanted to say
Let's go.
that I wanted to say that we took some advice that taking some advice from super agents can lead us to to better treatments for Alzheimer's disease. And I'd like to leave you with the knowledge that for some older adults, as we talked earlier, age-related cognitive decline is not inevitable. We saw in these examples that there are superagers, and the superaging brain looks more like 20 years old brain than 70 years old peers. So yeah, having a youthful, super sharp brain is possible in older age, and dementia is not inevitable either. I think that if we can understand with brain imaging and other research what makes super agers special and continue to maintain these youthful brains despite their age, I believe that's the first step towards understanding resilience to aging.
Hmm.
And at this point, let me stop sharing and pull up the slide about the MADRC registry if people want to reach
There you
out.
go.
In my other browser, did I stop sharing? I think I did. Hmm. Okay, I got it. Okay. Do you see okay in the slide?
Yep, yep.
Is it clear?
Um, so hold on, yeah, no, go back. There you go. Leave that there. Leave that there.
Okay.
Hold on. Do. My grandmother. Oh, yeah. We all, yeah. Staying active, as Alexandria said, staying active is what you want to do, what you want to do. So a question that always comes up whenever we're talking about MRIs in the community is, excuse me, does insurance cover the cost of getting a PET scan or an MRI?
So the insurance covers the cost of the amyloid PET, but does not cover the cost of the tau PET. I know that there are efforts at Mass General and other institutions to have the tau PET also being covered. But people can sometimes what is happening is that when an order is needed for a tau PET and it's not being covered in the clinic, because the clinic is so much interlinked with research, and we share the scans, the researchers, the neuroscientists will share the scans with the clinicians to better understand the disease of that particular person. We may enroll someone into a study, a research study that includes tau PET,
There you go.
and yes, and they were not going to pay.
In fact, correct me if I'm wrong, but don't most studies pay
Yes.
someone to participate?
In all of our studies, yes, we reimburse our participants for their invaluable time and effort to being involved in research, because this is time commitment, particularly in clinical trials. It's a lot of time commitment, but also in other studies like observational studies that include only brain imaging. People will have to come and be part of the whole MRI preparation or the PET preparation. So it's time commitment and effort, and we appreciate all this effort. And yes, we reimburse the participants for the participation and the travel and other related costs.
So here's another question with the MRI. Someone asked this earlier in the chat, and kind of sort of in a way. So well, first of all, do you share the information with about what's in the MRI? Someone who signs up for a research study and they go through an MRI, do they get the information? In other words, do you tell them what you found?
So we are working towards disclosing the results to the participants that are symptomatic. For those participants, if we see something unexpected in MRI, we will then communicate with the primary doctor, and we will communicate with the participant and be in contact with their neurologist or the primary care physician to discuss this.
So if you find something you weren't looking for,
Yes.
Then which is another good reason to be part of this study.
I would say that in many of our research studies, it can help you monitor. It has an additional measurement of how the brain structure, the brain function, and where for those participants that are symptomatic, where the stage of the disease is, or what is the efficacy of the treatment they are being involved. So it helps both the participant and the scientist to better understand the disease, but also the participant to get more information about where they are, at which stage they are.
So the one that, all of these studies are amazing, but I'm going to butcher the name, so I'm going to call it the brain massage one, the magnetic brain massage. How often, if someone enters that study, is that one time every 6 months, once a year? How often do they have to come in?
What study, Mike?
I'm
The
blanking.
coil? The one with the
Yes,
coil?
the coil. Yes, there you go.
The non-invasive brain stimulation. So the non-invasive brain stimulation study, it's a study that requires time commitment, because it's 10 days non-invasive brain stimulation for 10 days. And then there's a break of 3 weeks, and then participants will need to come back for 10 more days, because each participant is getting the active stimulation and the placebo stimulation. Everyone is getting the active and everyone is getting the placebo. We don't know, none of us know the order of it. If you are getting first placebo and then the active, or the other way around, but everyone gets the active stimulation. And so it is about 2 months if you consider also the break and the before and after measurements, because before the brain stimulation study, we do brain imaging, and we do brain imaging for two reasons. The first is because we want to see, when we stimulate, we want to stimulate the ideal target for that particular participant. And
Mhm.
to be able to identify that, we need the structural MRI and we need the functional MRI. We need to make sure that we are improving the right spot, which is connected to the regions of the brain that are supporting, let's say, language. Let's say a participant comes to our study with atypical form of Alzheimer's disease, who has symptoms, who has language impairments. We want to stimulate the network, the brain network, that it is important for language processing and language abilities. In order to identify that, we do the very first visit, we do brain imaging. We identify the brain regions in that particular person that are important for language, and then we use that information to develop a protocol of stimulation such that we target in a personalized way in that particular person the ideal target, the ideal regions. So that's one reason why we do brain imaging before the clinical trial. And the second reason is that we want to measure, as I saw earlier, the change in brain function before and after the brain stimulation. So in order to do that, we need a brain imaging before the stimulation and a brain imaging after a brain scan after the stimulation to compare before and after. So we use that for to measure the outcome of the trial.
Okay.
So
You know,
it is that's why I think that brain imaging it's an integral part of research and
Yeah.
of what we do as neuroscience in neuroscience.
all right. Siobhan, I see your hand, but before I go to you, a quick question. And I realized this. Where the heck are you guys located? Are you at the Brigham at the main campus, Charlestown?
We are, yes, we are. Our research is all in the Charlestown, Mass General Hospital, Charlestown, Navy Yard, 149 building.
Okay, all right.
We
Which
do
they'll
our
find.
imaging at the Martinez Center.
Yep, yep. Yep, the center. So, which they'll
Yes.
find out when they do the link thing and all that. Siobhan, you've got your hand up.
That I do. I just have a quick question. Just listening and watching, looking at the different images. Does like for a female, like going through like perimenopause and menopause, does that change the brain image of a thing that you're looking for? Like, does that affect what you're seeing? Like things like that. I'm thinking of like when you even get like a mammogram, sometimes it looks like something, but it's not, because depending on where the woman is in her life. So I'm just thinking, does that look different? And does that affect what you're looking for?
A very close colleague is actually studying this exactly question. He got funding for his research, and he's looking at exactly the changes that are happening with menopause and premenopause and menopause, and how that hormone levels affect brain function and brain structure, and if that is linked to a greater risk for Alzheimer's disease in women. That's a great question. It's a
Thank
research
you.
question actively being studied. I hope I
I'm
should
good to
have
hear
more
that it's
results.
actually.
I hope I
Sorry.
have more answer to that question when we meet next time.
Thank you. I'm happy to hear that actually it is an active study to see how that looks. So thank you for that.
In fact, we owe everybody a part two on the menopause topic. So let's get that done this summer, because Alexandra, we did a partner night a topic night on menopause, and I think we shattered the record for the most folks. And for me, it was educational and enlightening. I learned more that night than I ever knew. So we're gonna
That's great.
so tell your colleague we'll be calling.
Absolutely.
So they say no one ever hates you if you wrap early. And it looks like we've got no more questions. So, Alexandria, if there's one thing you want folks to remember about tonight, besides how to get in contact with you, and for everybody on Facebook, tell them once again how to get in contact with you.
So did I I'm not sure I didn't did I write my email in the
You
chat?
put it in there. Siobhan, does the chat show up on Facebook?
No,
Okay,
no,
so
the chat will not show up on Facebook.
Okay,
just
should I
say,
put I
no,
should just send my email in the chat?
I was gonna say uh just say what your email address is. Or your
It is
contact, how, because it
mm,
won't show up on Facebook Live, and we've got folks, shout out to everybody on Facebook Live. We've got folks on Facebook Live who can't see the chat. So,
okay, so it is A Touroutoglou at mgh.harvard.edu. This is
all
the
right.
email, and yes, and the office number is 617-643-6348.
Could you say that again?
617-643-6348.
All right. All right. So if there's one thing before we jump on our dance party, if there's one thing you want folks to remember, besides their phone number, if there's one thing you want folks to remember about tonight's conversation, which has been amazing, thank you so much. What would
Thank
be
you
that
for
one
having me. We really wouldn't know all the things we know about research, and so far all of our progress in Alzheimer's disease and in healthy aging and in successful aging and super aging without your participation in our research. So I want to express my gratitude for all of those of you who come and participate in our studies. It helps us advance science, and really helps us understanding the disease. As I said,
excellent.
it's not just science, it's about humans and about humanity. Thank you. I would be more grateful.
Alexandria, thank you so much. Thank you so much. My closing thought before we crank it up is I want to, you folks have heard me say this before. We need people to participate. I'm going to be direct. We need people of color to participate in dementia research because we are more likely to get this disease than our white counterparts. So we need to show up. We need to tell people. We need to not, I'm not saying don't be afraid, because it's okay to be afraid, but still show up and let's conquer this disease. People ask me, why am I in the world of Alzheimer's? So that I don't have to tell my grandson that his great-great-grandmother had dementia, because she's done so much more in her life than to have a disease define who she is. I want to thank everybody as always. Yeah, Purple Rain, you're amazing too, girl.
Thank you very much.
See, someone always sneaks in a question at the end. Melinda, take us home, girl.
Yeah, no, I just wanted to number because I didn't catch it.
The number,
Her number.
Okay.
it is 617-643-6348.
648. 64. Okay, 6348.
617-643-6348.
48. Thank
Yep.
you so much.
All right.
Thank you all
Okay.
very much for participating and all these great questions and discussion. I enjoyed and I really appreciated your support. Thank you.
And Alexandria, thank you so on behalf of everybody. And
Thank you.
anytime you want to come back, you let me know.
Thank you.
You let me know.
I would love to come back with more results.
When you're ready, let me know.
I will.
All right.
Thank you.
All
Have a
right.
good night.
Siobhan, take us home, girl.
All right, Mike has spoken. So I do want to thank all of the network coordinators here. Dr. Alexandra, thank you so much for a very informative presentation. First time being here, so everybody give her a round of applause. And definitely, you're always welcome to come back. Claudio, as always, I appreciate you. We had 187 of us on here tonight. There's
Oh,
so much
wow.
going on with all the nice weather. You could have been doing anything else tonight, but you chose to be here with us. So as always, I love you, and there's nothing you can do about it. But we are going to close out, like Mike said, with some music. If I can find the button. Where's the button?
Don't make me have to sing.
That I am not singing. We would run out of here quicker than anything. All right, so here we go. Come off of mute, everybody.
All right. Good night.
Thank you. Have a good evening.
Good night,
everyone.
Happy Mother's Day.
Happy Mother's Day, everybody.
Mother's Day. Good night.
Happy Mother's Day. Have a great weekend.
Happy Mother's Day.
Blessings to you all.
Thank you. Have a good night, everybody.
Everyone. Good night, everyone. Happy Mother's Day. Good night, everyone. Have a blessed evening.
Yep, and I will see you this Saturday at, thank you for not in the chat. This Saturday from 10 to 1, we will be at the Wilson Hilton for our resource fair. So if you want to see Mike Kincaid in person, come on down.
Be there for a resource Saturday.
Don't miss it.
Yeah.
You know what? In fact, I'm gonna do a sneak preview. Yeah.
Good night, everyone.
Oh, you can't see it.
Can't see it, Mike.
There we go.
Good night, everyone.
Raffle prize. Good night, everybody.
Bluetooth candle speaker.